Hi, Amirreza here. I'll be presenting our work titled Gene Network Connectivity Conveys Robustness in Gene Expression Across Individuals, Cell Types, and Species. This is joint work with Olga Sigalova, who is currently a postdoc at EMBL, and my supervisor Judith Zog, who is a group leader at EMBL. Okay. Living systems have a remarkable capacity to be robust against different sources of variations, such as genetics, environmental, and stochastic. In this work, we aim to better understand this property from the viewpoint of gene expression program. More specifically, we are interested in understanding the buffering mechanisms of gene expression. To be more precise, perhaps the main question is, what factors are controlling the shape of a gene's distribution? Since nearly 2 decades ago, a growing body of work has been extensively studying this topic under the similar names, including gene expression variation, variability, and noise. In particular, our previous study reveals genomic features that make a gene highly responsive to any type of variation. However, in previous studies, the connectivity of genes through the network is usually not taken into account. To start discussing our results, I briefly mentioned the setup in the scope of this talk. We consider data from GTEC, GWADIS, and HIPSCI projects to cover different tissues and 2 cell lines, namely B cells and pluripotent stem cells. Moreover, we consider residual CV as the main variable that we are interested in. In fact, to have any meaningful conclusion about the properties of coefficient of variation, CV, we need to make sure that it is statistically independent with the median, as there is a well-known dependency between these 2 variables in the gene expression data. We then take the mean of these residuals across tissues and cell lines to have an overall view of variation, and we simply call this variable variation. We also rely on the aggregated co-expression network across different datasets of a prior publication. Next, I will discuss some of our network results. Generally speaking, statistics of a node are related to average statistics of its neighbors. Moreover, the effect is stronger for the residual CV. In graph theory, indicators of centrality assign numbers to nodes according to their position in a graph. For example, we have eigenvector centrality. First, graph centralities of co-expression network are correlated with each other. More importantly, graph centralities are correlated with residual CV, and they are not much correlated with the median. As it can be observed, we have some Spearman correlation coefficients more than 0.5 between centralities and variation. Graph clustering or community detection methods reveal more separated groups for the residual CV rather than the median. Roughly speaking, these results can be achieved by using different graph clustering algorithms, although number of clusters might be different. Next, I discuss some of our prediction results. To the best of our knowledge, we built the most inclusive gene-specific regulatory features characterizing properties of gene promoter, enhancer, 3' UTR, and etc. In this talk, we focus on predicting mean residual CV. It is possible to predict the variation based on the sequence of the promoters. For this purpose, in summary, we used convolutional neural networks. As an ongoing idea, we are working on considering very long sequences using the recently published methods. Neural tangent kernel, NTK, demonstrated the best performance when we want to predict the variation based on our features. Moreover, this method can beat all champions such as random forest and gradient boosting. Actually, one of our contributions is popularizing this method in the genomic community. Incorporating the network into the prediction pipeline indeed improves the performance. Moreover, the improvement can be achieved by using a graph convolutional neural network model. Also, adding some additional features related to the graph structure can further a bit improve this result. Best performance in each track is demonstrated in the figure. As we can observe, we achieved more than 0.7 R² using our graph neural network model. As a final remark, it's notable that if we randomly permute feature vectors, we cannot get any meaningful R² using these methods. Okay, to summarize, our results indicate that gene co-expression network indeed plays an essential role in controlling the expression variation. There are actually many other results, several ongoing ideas, and different future directions that we can discuss during the conference. In the end, again, I would like to thank Olga and Judith. Without them, this work would not have been possible. Additionally, I thank Mattia and Eileen, collaborators of our previous paper. Thanks.